All right, that's the hard fade. So the first thing is Alex gets 0% on his DJing because there wasn't a soft fade out of that track. Good morning and welcome to Invertebrate 2700. Hooray! In month 10 of a pandemic. To start out, I love this. You got all of the ones that I got on the word puzzle. And you're demonstrating Zoom literacy, which is a surprise after 10 months of us being all in our rooms. Maybe not. So to start out, I see there's a couple of mentions in the chat that people are having a hard time hearing. Can you hear me? Any thumbs up or thumbs down?
I can hear you.
Super. Well, let's use that as a segue. Before we go much further, I said, I'm Alex Smith, and I'm going to be with you for on Mondays and Wednesdays and Fridays for the lectures. And why don't we start out and say hello to Sherry Hinks, who's going to be running the labs with you. Hello, Sherry.
Good morning. Good morning, everybody. Like Alex said, I'm going to see you in labs, and labs start next week. So I'm really excited to welcoming you. We've got some new activities planned. Remote offering is going to be different this semester, but I think we've got lots of fun things planned. And some hands-on activities, which will be great. Get you outside and get you doing things.
Cool. Yeah, we're actually really excited about what we've kind of come up with for this term. So we hope that we can either channel your excitement about the same thing or infect you with it. So I'm going to head back over to the PowerPoint and I'm going to clear all these drawings and I'm going to save them first because they were beautiful. And we will get underway. So I guess first of all, welcome again. Welcome to 2700. Welcome to 2021, the year when there'll inevitably be news. And part of the news that we're hoping to share with you is the news about the exciting world of the invertebrates, about which we're particularly excited. And we hope that you are too. You're here. That's a good sign already. But we understand that it's a year like no other. You guys were, if you were kind of probably in the majority, who were in first year enrolled last year, say roughly 10 months ago on the 11th or the 13th of March when everything shut down and we immediately pivoted to these digital environments. You're kind of the veterans of this. You've done maybe more of digital online learning than anybody. So take a second and recognize that and be proud of it. So put up your right hand in your room, pat yourself on the back. You're doing well. Hopefully, all of your classes, your professors, your instructors, we're getting better at talking and trying to be actively engaged with you in these online environments. And so we'll talk a little bit today about the course, this course, and some of the ways that we're going to try and interact. I'll give you a quick overview of where some of the details of the course, and then I'll give you my elevator pitch for why you ought to be excited about invertebrates if you're not already. Sounds like a plan? So one of the reasons that I'm excited about invertebrates is in this table. It's like the first table in the text, which we'll talk about in a second. And this isn't even the whole thing. It goes longer than this, but it essentially tries to capture that in this class, invertebrate zoology, invertebrate morphology and zoology, you're struggling with and being introduced to and celebrating 96% of the diversity of animal life on the planet, and that's of described species. The actual proportion is going to be larger because the majority of undescribed species that remain, that we share the world with, that we haven't recognized or described yet, they're invertebrates. And so at a minimum, 96% of the planet's diversity is included in this class. A maximum, it'll be, say, in the 98th percentile. So that's a lot. We recognize that. So 90%, so if we were to break that up and just talk about why or how much this of a lot this is, your course, your biology degree contains about 40 courses. So if that degree was scaled to the diversity of life, of animal life on the planet, in your final 8th semester in January of your 4th year, you'd get to take 2 classes that weren't this one. That to me would sound like an incredible degree and a great way to spend your time, and I'd love to spend that much time sharing and learning about all of these things with you, but it's probably not what you've set up in your course calendar or with your major advisor. But just, we understand, Sherry and I do, and all of the TAs, that this course contains a lot of things, and what we're hoping, what I'm particularly hoping, is that the a lot nature of this course doesn't become something that kind of like overwhelms you. It's something that you look at and you just kind of, you accept this is the diversity that we share the planet with. Let's be excited by it. And so one of the things that I, as a scientist who works with invertebrate diversity, I have to be super comfortable with saying, holy cow, I didn't know that. I probably shouldn't say holy cow as an invertebrate biologist, but let's say holy cephalopod. I didn't know that. That comes up again and again and again. I didn't know that fact about a morphological feature associated with a function. I didn't know that fact about a phylogenetic relationship. I didn't know that fact about a species or how to identify it. So we just have to be comfortable with that, and that's kind of exciting. That's the world that we live in. So welcome to the Nerd Zone. So in this class, today, what we're going to be talking about is the format. We'll test a little bit of the tech. You've already done a little bit of that, but we're going to try and kind of talk back and forth. I'm going to talk about the learning outcomes. I'm going to, as I said, give you my elevator pitch. I'm going to introduce you to the craziness that is, potentially introduce you to the craziness that is particularly my lecture. You see a door behind me here and a door over there. You may see other members of my family walking through. There's currently a first-year biology course being delivered or prepared to be delivered through that wall. And right over there, there is a kindergarten class happening. So, I'm Alex Smith. As I mentioned, you met Sherry Hinks, who's going to be running the labs, and the TAs. We're lucky to have a group of pretty cool TAs who have all taken this course, and some of whom have TA'd it before, and all of whom are excited about kind of meeting with you and figuring out how to share their enthusiasm for the invertebrate life. And there's also going to be other volunteers that you're going to meet in the labs. Volunteers are past graduates of this course who came away from it excited and jazzed, and not just enough to take Zoology 3700, but wanted to stay involved to share with you what they experienced in the year that they took the class. So there's a support structure here, let's say. So text the Zoom lectures on Zoom with me Monday, Wednesday, Friday. These lectures are recorded, and I'm going to be posting them to YouTube, and that link will appear in your course link where you find the PDFs of the lecture slides. The Zoom chat's not open, as you've seen already, but every day we'll have a specific chat in course link right from the main homepage of the course, and you'll see chat, and then you jump into the chat, and then there'll be a chat for each particular day. And I do that for a few reasons. One of them, the biggest one, is that you've got a record then on course link that doesn't depend on me uploading something, but everything that we talk about, the questions that you ask there, I can respond either verbally or with my fingers, and you can go back and check that out later on. It's also on the university system, which is better probably in the long run than on the Zoom system. We'll talk about the chat in a second about how I'm going to use it, because I've only got 2 monitors here. I don't have 4 or 5 or 6, so I'm going to put in some chat breaks where I go and check in to see how things are going. The lectures in general are there to introduce, to share enthusiasm, complement and extend materials in the text in the lab. I've worked kind of pretty hard to try and incorporate videos, a lot of photos and literature, up-to-date literature. Some of you, you've jumped down to the next point in that bullet, which is the lecture slide. The PDFs will be posted after course link, and that just has to do simply with the up-to-date nature of the lectures, particularly in this bananas year, is that those aren't going to be ready to be shared with you until after I've delivered them with you, okay? And another suggestion, just the last two, note-taking, and this is often tied in my mind at least, with the desire to have the lecture PDF beforehand. But note-taking is a really actively taking notes with your, while you're writing, maybe typing, maybe not. The literature suggests handwriting is even better and more particularly tied to memory and recall, because you're not as fast a writer as you are a typer. And because of that, you then have to synthesize the things that I'm saying. So that note-taking is critical to learning, and it makes you actively listen, and then you have to decide what's important. Now, of course, in this year, with synchronous, where we're all together, and asynchronous later on, you've got not just the PowerPoints that you can go back to and check and kind of cross-reference what your notes were from the times that we shared together, but you've got the actual lecture. So if you wanted to, you could go back and put me in like 5 times speed in YouTube, and we could go all the way through and share it with you again. So course link, if you haven't already been there, you'll see the chat box. That's where we're going to check in and meet together. It's also where, if there's announcements, those kinds of things. One example of an announcement might be in the event of a catastrophic internet failure at Chateau Smith, and we're shut down. And you don't see the lecture return immediately in the Zoom land, I would put my plans for you or for us or for me in that announcements on that day. So this is the chat check. So this is me now staring away from the camera, looking down at the chat. I see lots of people joining. I love invertebrates in all caps. That's basically, there's a tattoo right here. I see people greeting each other. I presume, what up, Raymond? And I'm going to presume is a positive thing. Awesome. All right. Smith returns. And we keep going. We talk about Zoom for a second. This is a New Yorker cartoon back in May when we thought the pandemic had been going on for a long, long time. That bright little light looking at, oh, look, everything's nice and clear. What a wonderful environment we have. And you can rest assured that whatever is kind of down here or over there, up here is pretty clean. And over there, chaos, just like it is at your house, I'm sure. So first thing that I'd like you to do is go up and tell me about where you are. So there's an annotate tool at the top of your toolbar. And if you select view options, you select annotate, you can select a stamp, and then tell me about where you are. Yeah, I see lots of people in the southwestern Ontario. Europe, amazing. Where is that, France or Spain? It's not Spain. He checks his eyes. West Coast. Oh, good morning. Time zones. Oh my goodness, time zones. Well done, everybody. So everybody who's not in the Eastern time zone, put your right hand, pat it up in the air, pat yourself on the back. Whether it's very, very late in the day or where are we there? Maybe early tomorrow. Well done. And outer space. I like it. It's kind of all of us. All right, I'm going to grab that and clear it. And let's say, for those of you in North America, zoom in a little bit. Show me again. Up and down the St. Lawrence. Alberta, BC. Where is that? Kapuskasing? Cool. Nice. All right, and I'm going to zoom in again. So here we are now looking specifically at the Ontario, a place to live, a place to grow. Sudbury, of course. Ottawa, Windsor, some of you down on the north shore of Lake Erie, it looks like. Up on the peninsula. Super good. Nice. I'm in Guelph. You can't really tell, I presume, from where we're sitting. I am in Guelph, and I'm excited, as I said, to be here with you. Let's go through one more time. Just another way for some Zoom kind of tech checking. Oh, skip that one. Forget it. This is a different one. Because when I think of, so this picture behind here, this is one of my favorite parts in the world. I miss being there a lot. This is Costa Rica and a cloud forest in Costa Rica. Show me about where you have visited. And if you don't find bits here, show me about bits where you want to go. The UK, Costa Rica, Dubai, Czech Republic, Egypt, Iceland. I think we all need to go to Indonesia. And Trinidad. And only a few to the Northwest Territories. And none of it. There's some places, this is a list I tried to drop of just places around the world. There's a lot of you to South Africa. That's super lucky. Well done. More to South Africa than to none of it. We have a big friggin' country. Those of you on the West Coast and outside of the Eastern time zone are aware of that. So lots to explore when we can explore again. And for right now, we're going to explore them digitally. All right, I'm going to clear those drawings. Thank you for playing along. And I'm going to check the chat again. Yes, okay, so there's a question about the dissection kits, and we will get to that. I'm going to talk about that in a second, because that's actually really important. Dissection tools in the activity kit, so we'll talk about everything that's in them. Sherry's going to talk about what's in them. I'm just going to encourage you to get them ASAP. Anything that you need for the dissections, the eventual dissections and the work is included in the kit. It's from the bookstore. Yes, it is. Yeah, so we'll talk about that activity kit, and Cape Town is amazing. Yeah, so then just a question from Aya Pakaria. Hello, good morning. Wondering about the lab sessions, and says on the week of it under the lab schedule, but in the email it says labs don't start until the 25th. Sherry can jump in and interrupt me if she's here, but I go by the email. Okay, and I'm going to jump back to the non-chat. So you met Sherry. These are a look at some of the TAs that are going to be helping us. Jennifer Gleason, Gavin Hosick, and Carolyn Tremblay. This is a look at some of the things that you might already be holding. This is your text, the 3rd edition. It's beautiful, it's heavy, and it's not super inexpensive, but part of the reason that we do that is because, as you said, as we said earlier, 96% of life on the planet, and it's the text that you use, or that we use for zoology 3700 as well. So it's a lot. If you just take this course, if you take both of them, it's kind of split over and it becomes half the price. You can get that at the bookstore. Via the bookstore. They have, at least when I looked yesterday, a couple, it said, of used copies still around. The other thing that's there, of course, is this activity kit. I encourage you to get that as soon as possible. This contains the arrangement of fun that that Sherry and team and we've prepared for you to use throughout the term for both lab and lectures. One of the things that is in there is a tiny little nasal strip. And we'll talk about in the near future how you're going to use those to sample some of the invertebrate diversity that lives on you and on me and on all of us. And so we're good. This is kind of our walking through the wilds this year. So the sooner you can pick that up, the better. The price that shows on the screen right now is different. There's a shipping fee that won't be applied, but you'll see that when you log in today. So this is a look at those little creatures that we're going to be looking at. This is with the nasal strips. This is a tweet of Sherry's from 2 years ago, maybe last year, maybe 2 years ago, when we were looking for Demodex mites. So these are some of the tiny things that we're going to be sampling and DNA sequencing from us. So we're kind of excited about that. This is a thing that is going to let us actually look at these animals really closely, really intimately, even though we're spread literally, as you saw, all over the world. One other neat thing, I think it's kind of neat, especially in this pandemic era, is that there is now a digital text option where you can buy the text or you can rent the text. So 180 days, that's $60 to get a digital copy of the text. 180 days takes you outside of when the final exam possibly will be. So if you're looking at this course and you're thinking and you're hearing me talk and you're thinking, I'm not too sure about this, I'm not too sure about the content, I'm not too sure about the people, this is kind of a low-risk way of investing. You can rent that text online. And there's a link there, and I will put that link in the chat, and it'll be posted in these lecture notes, and we'll put it up and make sure that you can find it easily. There's several other older texts, an old text that we'd used in the past, as well as some other people's view of the how an invertebrate biology textbook should be built, and those are on reserve. But generally, don't leave your house. So the office hours. What's an office? So office hours are going to be so I'm here with you Monday, Wednesday, Friday. On Friday, after the lecture, I'll turn off the camera and mute my mic for a second, go and get another coffee. You can ask me what number of coffee that is when I get back on Friday. And then I'll have the same Zoom link open for an hour, and we can chat. You can turn on your camera if you want. We can ask questions. We'll keep so that'll be the office hour. If that doesn't work, and I'll get a feel for your schedules throughout as we go, if that's not enough time. But if you want to ask me some other questions or private questions or whatever, that's my email. Send me an email and please put Zoology 2700 in the subject header. If we look at my email inbox right now, why am I asking you to put Zoology 2700 in the subject header? Because I get a lot of messages and I need to be able to sort through them somehow. And the ones that you're sending me are some of the ones that I consider important. So this helps me filter very quickly. So right now in my inbox, there's over 263,000 messages. That being said, if you don't get a response from me, maybe give me a grace period of 24 hours and then send it again. All you have to do is say, just re-upping this so that I can see it. Course link we use for archiving all of the materials, the PDFs, the assignments, of course, marks. We'll be doing a lot of quizzes there this year, announcements, discussions, and there's discussion boards there. So if you have questions about content, we'd really encourage you to go there first, talk about it with each other, and then come to the TAs, to Sherry and I, and ask about them then. There's also, we have a presence on Twitter. There's a Twitter account and a Twitter hashtag for this class where we'll use it and we'll tweet cool stories and photos and videos. You could tweet questions after or during the lecture, and that's not just for Sherry or I to answer or to comment on. You can chime in on them. If you use that hashtag Zoology 2700, we can compile them later on. Remember, if you have a Twitter account and you're tweeting, think about the fact that it's out there forever. If you've seen the news lately, there can be consequences to that kind of thing. And the 2 Ps, just be positive and be professional. There are some one of the things that I enjoy of all this social medias, is that this one is one that you don't have to belong to. You can just cruise it, and you can graze from it. So you can make your own account if you want, or you can just go in and take a look at these. So the department has one. There's a department, there's a group of undergraduates that has one called U of G Weeby IB that has one. And then each of actually the teaching team, we all have Twitter accounts there, so you can follow us to see what we're excited about. Follow or just drop in and check it out. There's going to be a lot of videos in class. One of the things that I'll try and use the discussion and the chat for is to put links for that I'm going to be referring to throughout the day. In addition to that, there's a long, literally long, like 26-hour long playlist of invertebrate videos that I will use or have used in years past in this class, compiled on YouTube. There's a link down there at the bottom. I think I might have put it in the chat for today already. And of course, the course link, which you've already seen. For the website, we talked about many of these things already, so I'll just jump through it. Oh, we didn't talk about a lot. So the lab report assignment tool, the gradebook exams and quizzes, especially in this pandemic year, we're going to be doing a lot more grading and a lot more testing on CourseLink. So that includes things that in the past we've called lecture midterms or a lecture midterm. So in place of that this year, and to try and get marks more evenly distributed and to have less stress associated with any one of these marks, we're going to be doing the lecture midterm quizzes in smaller chunks of the course. So you're going to be responsible kind of for less of the content at any one time. And we're going to be doing it with maybe a short answer question and a lot of true-false questions in that kind of a context on using the CourseLink quiz. That'll be the kind of thing that historically we give in a lecture time. So say you'd have 50 minutes to complete it. That's still what I'm writing. That's the kind of exam that I'm writing. And just to give everyone the benefit of the doubt, what we're going to be doing is keeping that quiz open for a long period of time. And so once you start, you'll have, I forget what number, 2 to 3 hours to do it. So I will never expect you to take 3 hours to do it. It won't take you that long. It's just once you start it, you'll have more time for doing it at your leisure. In the course syllabus, we talk a lot about these learning outcomes. There they are as a word. And what I like about them, and what I think about as we try and write them, is the active nature of the verbs in each of them. The most important 2 or 3 from the lecture perspective are say in the top. I appreciate the vast diversity of invertebrate taxa. If we can do that together, well done. And that's just critical upon critical to me, that we have more people in the world who are appreciative of and cognizant of the diversity, that which we know and that bigger chunk that we don't know. This is kind of the spread of the lectures and the spread of the marks throughout the assignments. And what I hope you'll notice there is that any one of them is not worth too much. I'm going to jump back to the chat. Oh my God, yes. We will not NFW will we be using Respondus. And if there's any way that we can help you if you are in a group that's compiling petitions contra Respondus, send them to me and we'll help circulate them. I'll help circulate them. Just a bananas, unnecessary program. A personal opinion. Yes, there's an answer from Sherry. Sorry about that, for what I said earlier. Labs start the 18th, so a week today, not the 25th. We'll be using the textbook in the course, or is it an additional... will we be using the textbook in the course, or is it just an additional source of information that we can use on top of the lectures? It is both of those things. We will be using it in the course, and it is an additional source of information for everything that we cover in the lectures. 96% at least of animal life. There's far too much for us to go through even in 36 lectures if we get that far. So there's a lot there, and it's there for you to kind of dive in and out of. It's an amuse-bouche. It's for you to sample as we go. Buying the textbook mandatory. Yes, it is. Okay, thanks. I think it's all included. Do we have to get all the parts of the kit? I was wondering about the lab sessions. Okay, I think I have caught up. All right. So I'm going to skip that for a second. We'll talk about some of the themes of the course. One of the big ones is unity and diversity, evolutionary relationship, who's related to whom, trends and changes in evolution, structure, morphological structure as it's related to function, and then the class as a whole serves as an introduction to concepts that are extended and you can dive more deeply into if you take Zoology 3700. So who are the invertebrates? This is not a figure from your text, but from a paper that we're going to be seeing a lot of, about 6 years old, by Casey Dunn and friends in the Annual Review of Ecology and Systematics. Who are the invertebrates? Well, they're basically the non-vertebrate animals. Hello. And this is one of the themes of my passion for studying invertebrates that I hope to share with you is the fact that there are, in the oceans at least, for example, 91 recent estimates, that there's as many as 91% of all species that remain to be described. The vast majority of those are invertebrates, 86% of the existing species on the planet. And this is, it's exciting. It's also a problem if you think about it in the context of global climate change and the anthropogenically forced climate crisis, where we are creating a selective environment with which we are losing species very rapidly, an era of mass extinction, where we have two competing rates, if you want to think of it that way. One rate is an increasing rate of extinction and a rate of species description that is not nearly as steep. Species description and understanding that's not nearly as steep. So a picture, if you can tell, that's from the Game of Thrones. You've taken vertebrate structure and function. Chordates have about 60,000, 61,000, maybe 62,000 species that have been described. That's a lot, and you survived this course, so pat yourself on the back. But this course, there are 60,000 of one named species of one family of beetles, the rove beetles. So just in one family of one class of one phylum, there are 60,000. There's as many species as there are vertebrates. So in a Game of Thrones perspective, when I think about vertebrates, I think about these Dothraki people saying, it is known. It is known. Vertebrates, it is known. We know. When I think about invertebrates, I think much more of egret. And we're in the invertebrate world, we're thinking of we're all Jon Snow. We're not the Dothraki anymore. We're Jon Snow. We know nothing. We know next to nothing. And we're excited by that. So why study invertebrates? This is into my elevator pitch on the way out of today's shared time together. Why do we study the invertebrates? Sometimes I get frustrated with that question and then I dial back and I'm like, okay, well, if people haven't had, there's kind of two things that can happen. One of them is that people haven't had the opportunity or inclination to do it. And the other is that many children from primordial ooze, we're taught not to have excitement about these things. And so I'm going to back off that head pounding on the desk and say, well, why do we study invertebrates? Because they're amazing. And so part of my job, our job, is to share with you why we think they're amazing, how that can affect your life as a human individual on the planet, and how there may be even, if you want to dial or always attach this back to what's good for humanity in the study, if humanity studies invertebrates and has a better understanding of invertebrate diversity, there's just an enormous toolbox potentially of design solutions, efficient design solutions that invertebrates through millions of years of evolution have winnowed away and been selected for, that we, if we understand them better, can extract and use to our advantage. And we'll talk about some of those design solutions later on. If you're wondering, even just one small example of that extension that I'm making there. Think about spider silk. Think about all sorts of ways that we use or we would like to use invertebrate designed morphological phenotypes that we can take advantage of and use them for ourselves to our advantage. And the other is opportunity and obligation. Like we talked earlier about the fact that you are a biologist. You are a biologist. Now at a time when we're losing species faster than any other time, certainly in the lifetime of anyone that you know, and faster than than has happened in tens of thousands of years. And so we have an obligation, I think, to try and better understand, and by understanding, protect the species that we share the planet with. And then opportunity. You have tools now that generations of biologists and invertebrate enthusiasts before you, from Miriam Sibyl to Charles Darwin to Alfred Russel Wallace, tools that they would have drooled over. Bates going through the Amazon and spending 8 years there. Tools that are on your phone, resident tools. So essentially having in your pocket a microscope and a way to record not just that you've seen something, but to record it moving. It's kind of an exciting time. And we'll talk about those adaptations or how those technological adaptations have changed and bettered our understanding of morphology and function, but also in terms of who's related to whom. Because phylogenies, as you talked about in 1070, as we talked about in 1070, if I was there with you, they're a hypothesis, right? And they're built with the best of evidence that we have available at a time. And in any particular era, those technologies change and are added to. And so the fact that we now have phylogenies that we can build not just with morphological features estimated using line drawings or estimated using photographs or estimating using scanning electron microscopes, but we have three-dimensional models that we can construct. We have heat-based imaging. We have not just DNA or the inferences of DNA, but we can sequence entire genomes and figure out who's related to who. With all of these things, we can go out into the field, into the deep and profound nature parts of the ocean, and film and record and see behaviors. You have an opportunity different than any other era of biologists ever. So why study invertebrates? And it's partially because this tortiere analogy, Ed Wilson, or E.O. Wilson, who's kind of the progenitor of the term biodiversity, the contraction, he took biological diversity and crunched it together. So we saved time. So thanks, Ed Wilson, for saving us some time. When we think about biodiversity, these numbers are not, they're all higher than that now. These are numbers that were known 40 years ago. The trends are more or less the same. The slices in this piece of pie are more or less the same. So tortiere, my French-Canadian mother, we eat a lot of tortiere, delicious tortieres, particularly at Christmas and at New Year's time. And if this animal, all of the animal diversity of life, if we thought about it as a tortiere, as a meat pie, why do we care about invertebrates? Because the meat pie of life tastes like invertebrates. We need to understand them if we had a hope of understanding diversity at all. Do pie analogies not help you? Shakespeare, Dickens, J.K. Rowling. How good would you, if you were responsible for sharing your enthusiasm for Harry Potter with a next generation of reader, how well could you do that if you only knew 2 or 3% of the characters in the book? You might do pretty well if by chance one of those characters was Harry or Hermione. But if they weren't, 2% of the characters, if you got one of the Malfoys or one of the lower end Death Eaters, well, not so good. So understanding more of this morass, these characters that we share the planet with, will lead to a better understanding of the play, of the story, of life on our planet. Think about it also in terms of biomass. So that last picture was about diversity, the number of taxa, or the number of species in that case. We also need to understand invertebrates because they are the vast amount of weight of animal on the planet. That's not saying ants alone equal 42% of the biomass of but invertebrates or insects in that case. Annelids, an enormous, this is always a surprising proportion, I think, to people, is how much of the planet is jelly. And Casey Dunn, who wrote that, or the author of the phylogeny that we're going to come to and you're going to see a lot of throughout the year, he made an analogy when he came here a couple of years ago to give a talk that if life from another planet were to arrive on our planet, by chance they ought to the first thing they'll bump into is the ocean, just because that's the majority of the surface area of the planet. The next thing that he would be most certain of is that the first life that they would bump into would be gelatinous. It'd be a cnidarian or a ctenophore. So maybe that's not how you currently operate thinking about the world in terms of the predominance of and importance of the jellies. There's another reason, this is an obligation, I think, is that if you're thinking at all for careers in terms of conservation or management, there's an enormous taxonomic bias towards our understanding of conservation and conservation biology, a bias towards vertebrates. If you look down at the bottom of this graph, this is the number of papers on the y-axis of conservation science, and take a look at the top 4. The vast majority of conservation papers are written about vertebrates, about fish, about or about amphibians and reptiles, about birds, about plants, and then we kind of start getting into some of the invertebrates later on. This is a problem if the umbrella effect in conservation in biology, I suppose as a modeling system, doesn't work. What's an umbrella effect? Well, it's when you say, we don't know about everything. We know about one thing very well. And that's our umbrella. And under that, if we make conclusions about that species, about which we know a lot of things, we hope that those conclusions extend to all the other things about which we know very little. An umbrella. The problem with that is there's just so many different ways of living in the world, from symmetry, whether you're bilaterally or radially symmetrical, to whether you're a warm-blooded or a cold-blooded organism. And the vast majority of the life is not, certainly is not warm-blooded and is not mobile like most vertebrates are, nor is perhaps even bilaterally symmetrical as we are. Most of the world doesn't see the world the way that we do. They don't see it at all. So to use I see as a synonym for I understand is just a disservice to most of life on the planet. They see the world through smelling and tasting. Most of the world does. So there's a bias here, a taxonomic bias that we're going to do our best to correct, at least to identify. Some other reasons. Health and disease. Why are we interested? So we're going to talk in a few weeks or in a few lectures about Toxoplasma gondii, Toxoplasmosis gondii, which can affect up to 95, is some estimates, 95% of human populations. And some have argued that this infection dramatically alters our behavior. And in a way, our behavior that can change our behavior dramatically, and that might not be due to us consciously changing it. It might be we're puppets being manipulated by parasitic infections. We think about cures to different diseases. So really from a human perspective, and there's recent research that suggests that a nematode contains an anti-inflammatory protein that could be a cure for asthma in humans. Disease so health and disease, mosquito species vector 600 diseases that can infect up to 700 million people a year. And then medical procedures, ways that we can benefit from, just like the leech example, the nematode example, in leeches that contain a protein that when applied to surgical procedures can increase the success of limb reattachment surgery by up to 80%. Why else might you want to argue to the family or those that you're sheltering in place with why you're taking this class? Some of them might be prone to economic arguments, ways that we take behaviors of different or features or functions of different organisms or groups of organisms, communities, and apply an economic value to them. And when people have done that for just one taxa, one group of beetles, the dung beetles, who actively decompose cattle feces in rangelands and pastures in one part of the United States of America, they found they estimated that service that to be worth a total of half a billion dollars. From an artistic perspective, we know that even 10 years ago, that invertebrates inspire modern art, music, multi-billion-dollar movie industry at a way that's disproportionate to many other things. Now, there's some direct examples, old ones like Bug's Life from Pixar and B-Movie, but then other ones where you might, so even older, but Alien. So practically any horror movie that you can think about that involves a creature, that creature was thought about by a person who'd studied invertebrate biology and taken one aspect of an invertebrate's behavior and then either scaled it up and made it big, or scaled it towards our species and attributed that feature to us. Pollination. More than $210 billion a year is the estimated economic value of pollination. Pollination occasionally contributed to by some vertebrates like bats in parts of the world, but almost exclusively done by insects. If it's done by animals, it's done by insects. And then if you think about this value was a bit of a surprise to me, but as we talked about Casey Dunn's example of the spaceship, the new arrival to our planet, arriving on the planet Earth, running into the ocean first of all, $40 billion. This is again about a 5-year-old estimate. That's the value, the global value estimated by the FAO for the sale of crustaceans. So one particular group of arthropods in the ocean, your shrimp, your etc. And so $40 billion. So these are not just esoteric pie in the sky kind of ivory tower concerns where what we need to know more about the species because Smith said they were like the players in the play. It's like, no, we need to know more about them because of health and hunger. If you're interested in who's eating whom and the players in the play, this is the invertebrates are what literally make environments. So reef-building coral species, polyps that are building three-dimensional structures that serve as a habit for most species of coastal fish. Not just most species of coastal fish in Australia, but most species of fish are dependent upon the structure, the habitat, their environment being created for them and maintained for them by invertebrates. So global reefs then, accounting for not just billions any longer. So we've left millions, we've left billions. We're now into trillions. $6.7 trillion of the animal global economy, and 17% of the protein we eat, we humans globally, is attributed towards coral reefs. 70% of life on the planet is probably parasitic. However, we know probably only 1, maybe 5% of the diversity of parasitoids or parasites. Parasitoids there's pictures here of caterpillar species and then a species of little innocuous wasp that inhabits one species of caterpillar. This was a case where in my lab we worked on this species of wasp that we thought was exciting because it ate a lot of things. And as soon as we started to poke at it a little bit more, what we found is that it was not a wasp that ate 36 different kinds of caterpillar, 36 different species of caterpillar. It's 35 different species of wasp, morphologically nearly indistinguishable one from another, but biologically incredibly different. And this is important towards biocontrol. If your family or the parts of the province or the world that you're from has come out as works in agriculture or in forestry, you know about the importance of biocontrol, where you use one species, targeted species, to knock down an outbreak of generally a lepidopteran, a butterfly or moth caterpillar that's defoliating a crop or a landscape. And then if you're at all interested in the understanding of food webs, of who's eating whom, this is invertebrates that historically, and you saw this back in 1070 where we talked about the Antarctic food web, these big black boxes that would just say plankton. And there could be dozens, hundreds of species in that black box that we don't actually know who's eating whom because we don't know the players in the play. So much to learn, so many jobs, so many areas that you can make yourself a global expert in them. And then diversity and unity. So this insect parasitoid that I talked about earlier, I think it's one of the most common and least understood forms of life on the planet. So an egg laid in or near a developing insect larva that then burrows inside and develops within and bursts out. If you're looking at this and if you've seen the movie Alien or Aliens, this is where that idea came from. Coevolution, the idea that as species go extinct, so do their parasites. So one of the things, that's a California condor, so a vertebrate example. Ooh. But the California condor, when we took all the California condors out of the wild, helpful veterinarians made species of mite go extinct because they cleaned the condors that they brought in from the wild and unknowingly made species of mite that had not harmed the condor but had co-evolved with them for millions of years go extinct because those last habitats were then fumigated. And then obvious differences, this is diversity and unity, so despite obvious differences in form and function, we have the same requirements and for energy and raw materials, and we all respond to those same physical conditions in generally the same way. The same physical rules apply. So diversity, but unity. And again, unity in that, despite obvious differences, the rule sets that build us are the same. And then that allows us to make comparisons across and between taxa. So this is this is the last slide. And this is the done phylogeny. This, I'm hoping, will become not a scary phylogeny for you because we're going to see it a lot. There's only 32 things on the tips here. These are all the animal phyla. So we'll talk on Wednesday about how we're going to talk. So we'll remind you about some phylogenetic terms about who's related to whom, and we'll start diving into some of these examples. So I'm going to jump back over to the chat for a second. And I'm going to read through the chat for a bit. I'll be there for a bit trying to answer questions if there's anything that's not answered. I'm going to make my computer start recording or transmitting this recording to the YouTube. And I'll say, welcome. It's going to be a crazy year, inevitably, and we're going to try and get through it together. And it's our job to help you get through it in as best the ways you can. So welcome to Zoology 2700. I'm excited to be here, and take care, and we'll see you on Wednesday.